As we continue to talk about the cell membrane, I want to make just one comment about photosynthetic membrane. All membrane has characteristics in common. All membrane contains phospholipid and proteins. But photosynthetic membrane is a little more complex. Now remember, photosynthetic membrane is found in chloroplasts, that organelle that we find inside eukaryotic cells. But photosynthetic membrane is also found in prokaryotic organisms like our microbes. The difference is that in prokaryotes, like bacteria and archaea, they don't have any chloroplasts, so their photosynthetic membrane is their cell membrane. Their cell membrane has to do the work of photosynthesis. So what makes a membrane a photosynthetic membrane? The answer is pigment. Now, if you're not familiar with what this word means, the word pigment just is a molecule that is capable of absorbing light of a particular wavelength. If you recall from our discussion of microscopy, there's a big long spectrum in nature of electromagnetic radiation. It runs all the way from gamma rays at one end up to radio waves at the other end. And each form of electromagnetic radiation has a wavelength associated with it. The shorter the wavelength, the more energy that form has. So, a pigment molecule, and there are many different pigments, but a pigment molecule is capable of absorbing light of a particular wavelength. In photosynthetic membrane, the primary pigment is called chlorophyll. And you've heard that, I'm sure. Chlorophyll is what gives photosynthetic cells their very characteristic green color. So that's what the artist has drawn over here on the right-hand side of the slide. They've drawn a double membrane chloroplast, and they've shown some structures inside. We'll talk about chloroplast structure when we get into the eukaryotic organelles. But for now, know that what gives a chloroplast its green color is the pigment called chlorophyll. And it's chlorophyll that is the primary conductor, if you will, of the process of photosynthesis. Down here on the bottom of the slide, you're looking at some photosynthetic microbial cells. Each one of these little circles is actually a cell, and these cells have arranged themselves in these long linear sort of filaments. This is a particular type of microbe called cyanobacteria, and they have this characteristic of forming these chains out in the environment. So it's pigment that gives membrane a photosynthetic ability. It's pigment that helps capture the light from the sun and extract energy so that it can make carbohydrate. That is what happens during the process of photosynthesis. On this slide, on the right-hand side, we can see an artist's drawing of, again, a little piece of a cell membrane. A little rectangular chunk of a membrane has been drawn here. And as we said a minute ago, these are phospholipid molecules in a bilayer with the heads drawn in blue facing the outside of the cell and the inside of the cell where the water is, and the tails drawn in yellow facing the interior of the membrane. Now the difference in this drawing is that the artist has also drawn in proteins, And that's what all these strange looking globules are that are sort of floating around in this membrane. These are proteins. And remember, membranes are full of protein. The other thing we can see in this drawing are these strange chains. They're facing the outside of the cell. These are actually short chains of carbohydrate that are coming off of some of these proteins and some of the phospholipid heads. And we'll talk about those when we talk a little bit more about cell identity. Remember, what holds phospholipids together in a membrane is not covalent bonds. There are covalent bonds that build each individual phospholipid, but when individual phospholipids come together in a group, they spontaneously form a bilayer. And that's because of their chemistry, because their heads are hydrophilic and their tails are hydrophobic. They're held together by weak attractions. And that's what allows membranes to be very fluid, very flexible. The proteins that we find inside membranes are also held there by weak attractions. They're sort of floating in the membrane. It's a very flexible structure. It's a very fluid structure. It's what we now refer to as the fluid mosaic model. Now, for a long time, scientists didn't really know exactly how membranes were structured. We needed to develop more advanced microscopy techniques and other techniques in the laboratory in order to know exactly how membranes were structured. We know, for example, now that this is a very fluid structure, hence the fluid part of the name, and that it's sort of studded with these proteins. And that's the mosaic part of the name. Just like an artistic mosaic that you might find in an art museum. These proteins look like they're sort of randomly just placed within the canvas of the phospholipids. It has a mosaic look to it. And that's why we refer to membranes as having this fluid mosaic structure. Now, a couple things to know. Phospholipids within one layer of the bilayer. So we have this layer, and then we have this layer. And together they make up a bilayer. The phospholipids within one layer, or what we would call a monolayer, one layer, they are moving around. They're mobile. Remember, they're not attached to each other. So they are literally sort of bouncing around within that layer. They don't bounce between layers, but within one monolayer, the phospholipids are moving. And within this monolayer, the phospholipids are moving. And these proteins, some of which span both layers, they're also moving around. Now, this can be kind of hard to wrap your head around. It can be kind of hard to visualize. This is the analogy that I like to use. Imagine in your mind's eye that you're looking at a swimming pool. And now imagine that you're going to dump a whole bunch of ping pong balls into that swimming pool. In fact, you're going to put so many ping pong balls in there that the whole surface of the water is covered in ping pong balls. What you're looking at as you look down at that pool is the same as the surface of this membrane, the outer surface of this membrane. So these blue heads are all the ping pong balls that are in your pool. Now, you know that those ping pong balls are not sitting rigid next to each other. They're sort of waving up and down as the water in the pool moves. The same thing is happening with a lipid bilayer. There's movement, there's fluidity to the membrane. Now, Imagine that you take one of your ping pong balls and you paint it red. And you put that ping pong ball down, let's say in this corner of the pool. And then you go inside and you come back out a couple hours later. That red ping pong ball isn't going to be right here anymore, is it? It might be over here. It might be over here. It's not attached to anything. It can move, and it will move. It'll float, sort of bumping in between these other ping pong balls until it, reaches another location. The same thing is happening with each one of these phospholipids. They are moving around across the surface of the pool of this membrane. Now, if we take the analogy a step further, imagine that you have a ball, maybe it's a beach ball, and you toss it into your pool that's covered in ping pong balls. That would be like maybe this protein right here in purple. Now, if you put the beach ball right here, if you placed it right here at this spot in your pool, if you go in your house and come back out a couple hours later, that beach ball is probably not going to be here anymore, right? It probably would have floated through the ping pong balls to some other location in the pool. The same thing is happening with these proteins. They are moving, they're traveling in this fluid mosaic membrane. There's another type of lipid that we have to talk about now that we find in our lipid bilayer cell membranes. We said phospholipids are the primary lipid that you find, but there's another kind of lipid called sterol that is very common in cell membranes. Now, sterols are very unique looking molecules. You can see one drawn over here on the right side of the slide. They contain 1, 2, 3, 4 ring structures in them. So all steroids have these 4 big rings in them. Remember, when you see a structure drawn with lines like this, and there's no atoms drawn in, there's no C's or H's or O's anywhere. Everywhere there's a point, that's where a carbon is. So these are carbon containing rings. And sterols have these 4 rings in this structure. These are large molecules. They're bulky. And when sterols are inserted into a membrane, they make the membrane stiffer. They make it less fluid, less movable, less mobile and flexible. Now you might ask, why would a cell want to make its membrane less fluid? And the answer is that stiffer membranes are harder for molecules to cross. So when a cell loads up its membrane with sterol, it's making its membrane less fluid, and it's making the membrane harder to get across. So it's harder for molecules to pass from the inside of the cell to the outside, or from the outside of the cell to the inside. And for certain cells, that might be important. That might be protective, for example. Now, animals have a certain type of sterol called cholesterol in their membranes. I'm sure you've heard of cholesterol. Plants don't have any cholesterol, but they have another type of sterol. It's called phytosterol. Our prokaryotes, most of our microbes, bacteria and archaea, they don't have either one of those. They don't have cholesterol. They don't have phytosterol. In fact, prokaryotic cells, remember that's bacteria and archaea, those cells don't have fully formed true sterol molecules in them. Instead, they have sort of a primitive precursor to the sterol, and it has a name. Those molecules are called hopanoids. They're still very bulky. They have ring structures in them, but they're not true sterols. They perform the same function, though. When a prokaryotic cell puts a lot of hopanoid into its membrane, the membrane is going to become stiffer. It's going to be less fluid, and it's going to be harder for things to get across. So there are differences between prokaryotic membranes and eukaryotic membranes, but fundamentally they're the same. In other words, all cells, whether they're prokaryotic or eukaryotic, all cells have a membrane that is a bilayer. It is made out of phospholipids and proteins. It has that fluid mosaic structure. It has hydrophilic heads that face the water outside the cell and inside the cell. And it has hydrophobic tails that are tucked in the middle of the membrane away from the water.